Everyone, and welcome to my YouTube channel Perfect Lab. In today's video we will learn about the Simmons Citrate Test, a simple but important biochemical test used to determine whether a bacterium can utilize citrate as its sole carbon source. We will also learn about the test principles, the composition of the medium, the step-by-step procedure, result interpretation, and the significance of these tests in bacterial identification. So let's get started. Start our introduction by looking at what Simmons Citrate Test is. The Simmons Citrate Test is a biochemical test used to determine whether a bacterium can utilize citrate as its sole carbon source and ammonium salt as its sole nitrogen source. The test is commonly used to differentiate members of the enterobacterial side and other gram-negative bacteria. And the test is performed using Simmons Citrate agar, which contains citrate as the only carbon source and ammonium dihydrogen phosphate as the only nitrogen source. And bacteria capable of using citrate grow on the medium and produce alkaline byproduct to result in a characteristic color change of the medium. Simmons Citrate Test principle. The Simmons Citrate Test is based on the ability of certain bacteria to utilize citrate as their sole carbon source and ammonium dihydrogen phosphate as their sole nitrogen source. Bacteria that produce the enzyme citrate permease can transport and metabolize citrate to produce alkaline byproducts such as sodium carbonate and ammonia. These compounds increase the pH of the medium, causing the bromothymol blue indicator to change from green to deep blue to indicate a positive citrate test. If the organism cannot utilize citrate, there is no growth, the pH remains unchanged, and the medium remains green to indicate a negative citrate test. So let's see how this works. On my left, we have a Simmons Citrate agar slant, which is green before inoculation, and the only carbon source in this medium is citrate. Now, most bacteria cannot use citrate because they lack a transport protein called citrate permease. However, citrate-positive bacteria produce citrate permease, which allows citrate to enter the bacterial cells where it can be used as a carbon source. And once citrate enters the cell, the bacteria metabolize it to obtain energy. And during this process, alkaline byproducts such as sodium carbonate and ammonia are produced. And these alkaline compounds increase the pH of the medium, making it more basic or alkaline. So, as the pH of the medium increases, the bromothymol blue, which is acting as the pH indicator in the medium, changes color from green to deep blue to show or to indicate a positive result as shown here. So, in summary, bacteria that can utilize citrate produce the enzyme citrate permease, which allows citrate to enter the cells. The bacteria then metabolize citrate to produce alkaline byproducts such as sodium carbonate and ammonia. And this alkaline compound will increase the pH of the medium to cause the bromothymol blue indicator to change from green to blue. Therefore, a blue color indicates a positive citrate test, whilst a green color indicates a negative citrate test. An additional information we must note about the Simmons Citrate agar is that it's a selective medium that tests an organism's ability to use citrate as the sole carbon source. So now let's look at the Simmons Citrate agar composition. So the first component is sodium citrate that acts or serves as the sole carbon source to differentiate citrate utilizing bacteria by allowing only organisms that have the ability to use citrate as their sole carbon source to grow. The second component is ammonium dihydrogen phosphate that also serves as the sole nitrogen source to allow bacteria only capable of utilizing citrate and ammonium to grow. The third component is dipotassium phosphate that serves as a buffer to maintain a stable pH in the medium. The next is magnesium sulfate that acts as a cofactor, which is essential for enzymatic activity. The next component is bromothymol blue that acts as a pH indicator and turns blue if citrate is utilized. And the last component is agar, which serves as a solidifying agent to solidify the medium. Let's look at the step-by-step procedure for performing Simmons Citrate Test. The first step is to label the Simmons Citrate agar slant with the sample identification, date, and other necessary information. The next step is to flame to sterilize the inoculating loop and then allowing it to cool before using. The third step is to use aseptic techniques to pick a small amount of a well-isolated colony from a fresh bacterium culture as shown here. The fourth step is to inoculate the slant by lightly streaking the surface of the Simmons Citrate agar slant. And one important information that we have to note here is that we are not supposed to stab the bud, and we should also avoid using a heavy inoculum as this may produce false positive results. So this is a picture showing the correct way of inoculating your agar slant by lightly streaking the surface of the Simmons Citrate agar. The next step is to incubate the inoculated tube at 35 to 37 degrees Celsius for 24 to 48 hours. And what you also have to note here is that some slow-growing organisms may require up to 72 hours to show a positive result. So let's look at the quality control in the Simmons Citrate test. So for the quality control, we use a known bacterium that tests positive in the Simmons Citrate test and a known bacterium that tests negative in the Simmons Citrate test. So for the positive control, we use Klebsiella pneumonia, which is a known bacterium that gives a positive test result in the Simmons Citrate test, and E. coli as a negative control that will give a negative test result in the Simmons Citrate test. Interpretation of Simmons Citrate test result. 1. A blue medium with visible growth after incubation indicates a positive test result, and this means that the inoculum or the bacterium here can utilize sodium citrate as its sole carbon source and ammonium dihydrogen phosphate as its sole nitrogen source. A green medium with no growth after incubation indicates a negative test result, and what it means is that the inoculum or the bacterium here cannot utilize sodium citrate as its sole carbon source and ammonium dihydrogen phosphate as its sole nitrogen source. A green medium with visible growth after incubation indicates a weak positive test result. And what we have to note here is that the Simmons Citrate medium itself is a selective media, so it will only support the growth of organisms or bacteria that can utilize citrate. Therefore, just a visible growth in the medium indicates that the inoculum or the bacterium here has the ability to utilize citrate as its sole carbon source. And also the medium has not changed to blue yet because the inoculum or the bacterium, being a weak utilizer of citrate, will not be able to produce sufficient alkaline byproducts to increase the pH of the medium enough for the bromothymol blue indicator to change color from green to blue as shown here. Let's look at some important notes or things to consider when performing the Simmons Citrate test. One is that heavy inoculum may cause false positive results. So it is not advisable to use heavy inoculum. So some organisms require longer incubation before becoming positive. 3. Growth without a color change should still be considered a positive result, and the test should be performed with a pure culture. The clinical importance of the Simmons Citrate test. 1. It is useful for differentiating members of the enterobacterial side, and 2. For distinguishing Klebsiella, Enterobacter, and Citrobacter species, which are usually citrate positive, from E. coli, which is typically citrate negative. So thanks for watching, and if you found this video helpful, please like, share, and subscribe to Perfect Lab. Leave your comments or questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video.